Chapter 3, Methods of Cognitive Neuroscience. Cognitive neuroscience stems from the principles of cognitive psychology, the study of mental activity as an information processing problem. Cognitive psychologists are interested in describing human performance, the observable behavior of humans and other animals. They also seek to identify the internal processing, the acquisition, storage, and use of information that underlies this performance. A basic assumption is that our perceptions, thoughts, and actions depend on internal transformations or computations. Cognitive psychologists design experiments to test hypotheses about mental operations by adjusting what goes into the brain and then seeing what comes out. Cognitive neuroscience is distinctive in the study of brain and behavior because it combines paradigms developed in cognitive psychology with methods employed to study brain structure and function. Two key concepts underlie the cognitive approach. First is that information processing depends on internal representations, or the cognitive symbol formed in the brain that represents an external reality. These mental representations undergo transformations. We usually take for granted the idea that information processing depends on internal representations, like beliefs, thoughts, concepts, desires, perceptions, and so on. Consider the word ball. Are you thinking of an image, a word description, or a mathematical formula? Each instance is an alternative form of representation and depends on our visual system, our auditory system, our ability to comprehend the spatial arrangement of a curved drawing, our ability to comprehend language, and our ability to comprehend geometric and algebraic relations. This figure here indicates a letter matching task, which was first introduced by Michael Posner in 1986. It provides a powerful demonstration that even with simple stimuli, the mind derives multiple representations as depicted in this figure. In the experiment, two letters are presented simultaneously in each trial. The participant's task is to evaluate whether they are both vowels, both consonants, or one vowel and one consonant. The participant presses one button if the letters are from the same category, and the other button if they are from different categories. Reaction time, the speed with which participants make their judgments, is plotted as the dependent variable. Response latencies differ significantly. Participants respond fastest to the physical identity condition, next fastest to the phonetic identity condition, and slowest to the same category condition, especially when the two letters are both consonants. The results demonstrate that we derive multiple representations of stimuli based on different aspects of the item used. Memory retrieval in daily life draws on a number of cognitive capabilities. Saul Sternberg in 1975 introduced an experimental task to explore a similar idea. In Sternberg's task, the job is to compare sensory information with representations that are active in memory using a series of letters. Sternberg postulated that to respond on this task, the participant must engage in four primary mental operations: encode, compare, decide, and respond. With encode, the participant must identify the visible target. With compare, the participant must compare the mental representation of the target with the representations of the items in memory. With decide, the participant must decide whether the target matches one of the memorized items. And with respond, the participant must respond appropriately for the decision made in step 3. The question was how to characterize the efficiency of recognition memory. The recognition process must work in one of 2 ways. A highly efficient system might simultaneously compare a representation of the target with all of the items in the memory set. On the other hand, the recognition process might be able to handle only a limited amount of information at any point in time. Sternberg found that the comparison process operated in a sequential or serial manner. Thus, reaction time increased in a constant or linear manner with set size, and the functions for the yes and no trials were essentially identical. In the memory search experiment, participants are not able to compare the target item to all items in the memory set simultaneously. That is, their processing ability is constrained. People can do only a certain amount of internal processing at any one time, and will also experience task-specific constraints. Exploring the constraints in task performance is a central concern for cognitive psychologists. Consider a simple color naming task, the Stroop task, devised in the early 1930s by J.R. Stroop. This has become one of the most widely employed tasks in all of cognitive psychology. The Stroop task involves presenting the participant with a list of words, then asking her to name the color of each word as fast as possible. This task is much easier when the words match the ink colors. This test demonstrates the multiplicity of mental representations and activation of a representation based on the word rather than the color of the word appears to be automatic. This is one test that aims to identify the mental operations that are required to perform cognitive tasks and explores the limitations in task performance. An integral part of cognitive neuroscience research is choosing the population to be studied. Study populations fall into 4 broad groups: animals and humans that are neurologically intact, and animals and humans in which the neurological system is abnormal, either as a result of an illness or disorder, or as a result of experimental manipulation. We begin this section with a discussion of the major natural causes of brain dysfunction. Nature has sought to ensure that the brain remains healthy. Structurally, the skull provides a thick, protective encasement, while the meninges layers surrounding the brain provide nourishment along with the extensive blood supply. Even so, the brain is subject to many disorders, and their rapid treatment is essential to reduce the possibility of chronic, debilitating problems or death. Such disorders include vascular disorders, tumors, degenerative diseases, traumatic brain injury, and epilepsy. Cerebral vascular accidents or strokes occur when blood flow to the brain is disrupted. An angiography is a clinical imaging technique used to evaluate the circulatory system in the brain and diagnose disruptions in circulation. This brain on the left is from a person who had an occlusion of the middle cerebral artery. The person survived the stroke. However, after death, a post-mortem analysis showed that almost all of the tissues supplied by this artery had died and been absorbed. On the right is a coronal section of a brain from a person who died following a cerebral hemorrhage. The hemorrhage destroyed the dorsomedial region of the left hemisphere. The effects of cerebrovascular accident 2 years before death can be seen in the temporal region of the right hemisphere. Brain lesions can result from a tumor or a mass of tissue that grows abnormally and has no physiological function. Brain tumors are relatively common. Most originate in glial cells and other supporting white matter tissues. With brain tumors, the first concern is not whether the tumor is benign or malignant, but rather its location and prognosis. Concern is greatest when the tumor threatens critical neural structures. Neurons can be destroyed by an infiltrating tumor or become dysfunctional as a result of displacement by the tumor. Also, abnormal pressure can be placed on brain areas and cut off brain supply, causing damage. Degenerative disorders have also been associated with both genetic aberrations and environmental agents. A prime example of a degenerative disorder that is genetic in origin is Huntington's disease. Genetic links for Parkinson's and Alzheimer's are weaker, although neurologists were able to develop a taxonomy of degenerative disorders before the development of neuroimaging methods. Diagnosis today is usually confirmed by MRI scans. In example, the scans depicted here demonstrate the brain of an individual with Alzheimer's disease on the left versus that of a healthy control on the right. These arrows indicate the growth of white matter lesions and the marked atrophy in areas of the cerebral cortex. Trauma can cause extensive destruction of neural tissue. Damage can arise from the collision of the brain with the solid internal surface of the skull, especially along the jagged surface over the orbital region. In addition, accelerative forces created by the impact can cause extensive shearing of dendritic arbors. In this brain of a 54-year-old man who had sustained a severe head injury 24 years before death, tissue damage is evident in the orbitofrontal regions and was associated with intellectual deterioration subsequent to the injury. The susceptibility of the orbitofrontal region to trauma was made clear by A. Hallbaum of Oxford, who in 1943 filled a skull with gelatin and then violently rotated the skull. Although most of the brain retains its smooth appearance, the orbitofrontal region has been chewed up. We will see how brain damage, particularly of the orbitofrontal region, affects behavior and social cognition in later chapters. Epilepsy is a seizure disorder in which recurring seizures are the main symptoms and are caused by an excessive and abnormal discharge of electrical activity from the neurons in the cerebral cortex. Types of seizures include grand mal seizures, petit mal, and absent seizures. Grand mal seizures involve total body convulsions and are also known as tonic-clonic seizures. Petit mal involves isolated body part convulsion and are also known as focal seizures. Absent seizures occur when a patient becomes unresponsive and has no memory of the occurrence. These are very rare. The figure demonstrates electroencephalographic recordings from 6 electrodes positioned over the temporal, frontal, and occipital cortex on both the left and the right sides. Activity during normal cerebral activity is depicted by the traces on the left, while activity during a grand mal seizure is shown on the right. Lesion studies either occurring naturally in humans or experimentally in animals, allow experimenters to test hypotheses concerning the functional role of the damaged brain area. The origins can be due to surgical ablation or removal or destruction of tissue, a lesion due to injury or disease like stroke, traumatic brain injury, or gliomas, or artificial lesions, where TMS allows for temporary artificial lesions. If a neural structure contributes to a task, then a structure that is dysfunctional through either surgical intervention or natural causes should impair performance of that task. Surgical interventions for treating neurological disorders provide a unique opportunity to investigate the link between brain and behavior. The best example comes from research involving patients who have undergone surgical treatment for the control of intractable epilepsy. The extent of tissue removal is always well documented, enabling researchers to investigate correlations between lesion site and cognitive deficits. Another example is in the use of treating Parkinson's disease, a movement disorder resulting from basal ganglia dysfunction. Though pharmacological agents are available, some patients do not continue responding well over time and may develop other symptoms requiring treatment. One widely used technique is deep brain stimulation, in which electrodes are implanted in the basal ganglia. This produces continual electrical signals that stimulate neural activity. Dramatic and sustained improvements are observed. However, it is unclear why the procedure works. Optogenetics methods may offer an alternative method in which clinicians can control neural activity, in that it causes the stimulation to change the interaction between different types of cells. This finding underscores that many diseases of the nervous system are not usually related to problems with neurons per se, but rather how the flow of information is altered by the disease process. We now turn to the methods used to study function, both at the neurologic and genetic levels in normal participants, including pharmacology, transcranial magnetic stimulation or TMS, and genetic manipulations. Besides being used in studies of chronic drug users, neurologically intact populations are used for studies in which researchers administer a drug in a controlled environment and monitor its effects on cognitive function. These drugs can either be agonist or antagonist drugs. Agonist drugs bind to a receptor and cause an effect, whereas antagonist drugs bind to a receptor and block an effect, thus interfering with activity of an agonist. An example of these types of studies is shown here in the study of dopamine, which is a key component in reward-seeking behavior. This study looked at the effect of dopamine on decision-making when a potential monetary reward or loss was involved. One group of participants received the dopamine receptor antagonist, haloperidol. Another received the receptor agonist, L-DOPA, the metabolic precursor to dopamine. Each group performed a computerized learning task in which they were presented with a choice of two symbols on each trial and had to choose between the symbols with the goal of maximizing payoffs. Each symbol was associated with a certain unknown probability of gain or no gain, loss or no loss, or no gain or loss. On gain trials, the L-DOPA treated group were faster at learning to choose the gains. than the haloperidol-treated group, whereas on loss trials, the groups did not differ, demonstrating that dopamine has an effect on reward-driven behavior. Transcranial magnetic stimulation offers a method to non-invasively produce focal or local stimulation of the human brain. The TMS device consists of a tightly wrapped wire coil encased in an insulated sheath and connected to a source of powerful electrical capacitors, which, when triggered, generates a magnetic field. The magnetic field passes through the skin and skull to create a physiological current that induces neurons to fire. When the TMS is placed over the hand area of the motor cortex, stimulation will activate the muscles of the wrists and fingers. TMS has become a valuable tool in cognitive neuroscience because of its ability to induce virtual lesions, as stimulating the brain disrupts normal activity. The method is appealing because it is safe and non-invasive. The figure on the right depicts TMS over the occipital lobe. When disrupting brain function in the occipital lobe with TMS, an individual's visual perception largely suffers, as indicated by the drop in correct identification of a stimulus. To understand the genetic component of cognitive neuroscience concepts, neuroscientists work with many animal models, seeking to identify the genetic mechanisms of both brain structure and function. A key methodology is to use genetically altered animals, referred to as knockout animals, which have specific genes knocked out or no longer present. This figure shows data from NMDA receptor hippocampal knockout mice, where the NMDA receptors in the hippocampus specifically were knocked out or removed. The study shows that the NMDA receptor knockout mice show an impairment in fear conditioning, indicating a role for NMDA receptors in the hippocampus, specifically in mediating the learning of the fear response. Several methods used to analyze brain structure utilize the differences in physical properties that different tissues possess. In this section, we discuss computed tomography, magnetic resonance imaging, and diffusion tensor imaging. Computed tomography has been a tool used since 1983 for structural imaging of neurological damage in patients. CT scanning allows for the reconstruction of three-dimensional space from compressed two-dimensional images, and the absorption of X-ray radiation in a CT scan is correlated with tissue density. By projecting the X-ray from multiple angles combined with the use of computer algorithms, a three-dimensional image based on tissue density is obtained. In this transverse CT image, the dark regions along the midline are the ventricles, the reservoirs of cerebrospinal fluid. It is very difficult to discern the differences between gray and white matter because they have similar densities. Magnetic resonance imaging scanners produce high-resolution images of soft tissue. MRI exploits the fact that many organic elements, such as hydrogen, are magnetic. In their normal state, the orientation of these hydrogen atom nuclei, an example protons, is random. When an external magnetic field is applied, the protons align their axis of spin in the direction of the magnetic field. A pulse of radio waves alters the spin of the protons as they absorb some of the radio wave energy. When the radio waves are turned off, the absorbed energy is dissipated and the protons rebound toward the orientation of the magnetic field. This produces energy signals, which are picked up by detectors. An MRI scanner can then construct an image based on the distribution of the protons and other magnetic agents in the tissue. The hydrogen proton distribution is determined largely by the distribution of water throughout the brain, enabling MRI to distinguish clearly the brain's gray matter, white matter, ventricles, and fiber tracts. Diffusion tensor imaging is a variant of MRI methodology that offers information about anatomical connectivity between regions, as it measures the density and motion of water contained in the axons. It is able to do this due to the lipid boundary created by myelin sheaths, limiting the flow of water in one direction of axonal communication. The development of electrodes and recording systems that can measure electrical activity within a single neuron or from a small group of neurons was a turning point for neurophysiology and related fields. This gave rise to the single-cell recording, as well as electroencephalography, event-related potentials, and magnetoencephalography. Electrophysiology allows for the recording of activity of single neurons in laboratory animals, and thus for the investigation into the response characteristics of individual neuronal elements. In studies with animals, an electrode is used to measure neuron activity in a slice of brain tissue kept alive in an artificial environment, or an electrode directly placed into the brain of a live animal, allowing for live recordings. In this example with the monkey, the activity of a single cell is monitored, and action potentials fire when a light stimulus is presented to the upper right quadrant, indicating the upper right as the receptor field for this cell. Single cell recordings in humans are rare. However, when surgical procedures are used in the cases of tumor removal or epilepsy treatment, intracranial electrodes may be inserted as part of the procedure to localize the abnormality in preparation of the surgical resection. In epilepsy, electrodes are commonly placed in the medial temporal lobe, where generalized seizures are most frequently focused. With electroencephalography, scalp electrodes measure the summed electrical activity of large populations of synchronously active neurons. They reveal changes in signal as a function of mental activity. Another approach focuses on how brain activity is modulated in response to a particular task. The method requires extracting an evoked response from the global EEG signal. The evoked response, or event-related potential, is a tiny signal embedded in the ongoing EEG that was triggered by the stimulus. By averaging the traces, scientists can extract this signal, which reflects neural activity that is specifically related to the sensory, motor, or cognitive event that evoked it. The visual evoked potential can be useful in diagnosing multiple sclerosis, a disorder that leads to demyelination. With demyelination occurring in the optic nerve, the electrical signal does not travel as quickly, and the early peaks of the visual evoked response are delayed in their time of appearance. Magnetoencephalography is a non-invasive pre-surgical mapping procedure. The electrical current associated with synaptic activity produces small magnetic fields that are perpendicular to the current. As with EEG, MEG traces can be recorded and averaged over a series of trials to obtain event-related fields. In this figure, the subject is undergoing MEG. The image shows a large tumor in the vicinity of the central sulcus. This technique measures spatial distributions of magnetic fields associated with large populations of synchronously active neurons, and it has good temporal and spatial resolution. However, the magnetic fields generated by the brain are extremely weak, so the device requires a room that is magnetically shielded from all external magnetic fields. The most exciting developments in cognitive neuroscience have been with imaging techniques that continuously measure physiological changes in the human brain that vary as a function of a person's perceptions, thoughts, feelings, and actions. The most prominent are positron emission tomography, or PET, and functional magnetic resonance imaging, or fMRI. These methods detect changes in metabolism or blood flow in the brain while the participant is engaged in cognitive tasks. They enable researchers to identify brain regions that are activated during these tasks and to test hypotheses about functional anatomy. PET and fMRI do not directly measure neural activity. They measure blood flow. When a brain area is active, more oxygen and glucose are provided by increasing the blood flow to that region. PET scanning allows metabolic activity to be measured in the human brain. In the most common form of PET, water labeled with radioactive oxygen is injected into the participant. As positrons break off from this unstable isotope, they collide with electrons. A byproduct of this collision is the generation of two gamma rays, or photons, that move in opposite directions. The PET scanner measures these photons and calculates their source. Regions of the brain that are most active will increase their demand for oxygen. Hence, active regions will have a stronger PET signal. fMRI measures the ratio of oxygenated to deoxygenated hemoglobin, as indicated by blood oxygenation level dependent, or BOLD, signal. It is an indirect measure of neural activity. And again, active brain regions means increased blood flow to that area, and increased BOLD signal. There are certain limitations to PET and fMRI. PET is constrained by the decay rate of the radioactive agent, and fMRI is dependent on hemodynamic changes. They both have poor temporal resolution as well, and correlation does not imply causation. For example, an area may be activated during a task, but not play a critical role in performance of the task.